Okay, so now I am recording. Reminder to everybody that the lab quiz is well, if you're in the morning lecture, it already opened today, and then there's another one if you're in Dr. Chu's, it'll open today at noon for 24 hours. Oh, Laren's from Laren's lab? Okay. I spoke to Dr. Chu yesterday. His is, he specifically said, could you please remind everyone if, so let me send an email to Laren and obviously. Okay, all right. Hopefully he's okay. And so I'll follow up on that too. And don't worry, we'll make sure things are squared away. So what are you asking then? Yes. Yes. Yeah. It'll be a 24-hour, when he opens it, it'll be a 24-hour thing again. That seems to be the fairest way to do it. So, okay. So, and then if you're in Dr. Chu's lab session, remember to take the quiz. It's apparently opened already at noon, since, right? Because you start lab at noon on Thursday. So you have 24 hours to take that quiz. So Dr. Chu, when I saw him yesterday via Zoom, he said, please remind the students. So, okay. I'm going to share my screen. Assuming I can find what I'm looking for. What's going on here? All right, I got something weird going on. Let's see if I can do it. All right, hopefully this will work. Okay, so hopefully Zooming students are just seeing one big screen. And so I'm putting this here. We've gone over this. This is the big picture idea, the pie chart look at carbon fixation. So we have the carboxylation step where the CO2 is coming in and reacting with the acceptor molecule RUBP. And then we also have the reduction phase. So by the time carboxylation is done, you end up with 2 molecules of PGA. And then reduction takes those PGA molecules and phosphorylates them with ATP, reduces them with NADPH, and you get your G3P triphosphate pool of molecules, some of which are destined to go out of the chloroplast and into the cytoplasm, so sucrose can get made, which is great. Some of it might get used for starch synthesis, and then it stays in the chloroplast if that's the case. But most of it will get recycled or regenerated so that you can have the acceptor molecule RUBP made again, so that you can keep taking in CO2 and doing this all over again. So you've been looking at this idea and the fact that Rubisco is that most important enzyme there that kicks everything off, where it allows CO2 into its active site and can react it with RUBP. Okay. However, we know that Rubisco is kind of a funky molecule, enzyme, because the C and the O word, part of the word is referring to the fact that it can carboxylate RUBP or it can oxygenate RUBP. And so that's what you're seeing on this figure. You're seeing both the both options here. So RUBP is right in the middle. And so on the left is what the plant is, really going after when it's trying to make sugars. It's taking in the CO2, comes into the active site of Rubisco, reacts with RUBP, and you get sugars being produced ultimately. On the right, though, they're showing the idea of the other option where oxygen might come into the active site instead of CO2. And then you do get one molecule of PGA in that process, which is great, but you get this funky 2-carbon phosphoglycolate. And that can't just smoothly fit into the Calvin cycle. Instead, it has to go through this whole other series of events and reactions called the PCO cycle. Okay, so when you do that, you do get a net loss of carbon in the form of CO2 that never seems to make it back into the chloroplast, and there's extra energy that's expended. So let's get into this a little bit. So here is just, if you want to see the actual chemical reaction process here, where RUBP is reacting with oxygen instead of CO2. And so you get one PGA molecule, which is being shown on the bottom, which is great. That can immediately go into the Calvin cycle. Good. Here's that 2-carbon phosphoglycolate on the top. It's not going to fit into the Calvin cycle. There's no enzyme ready to take that and just simply push it into the Calvin cycle. So that's not going to work. So what happens? This is what happens. All right. So this is an older textbook that I scanned a long time ago and put it in. To me, I like this one again because it's simpler than the one that's in your book. Not so cluttered, and I like the flow of it. So I'm going to teach off of this, but your book probably has something like that, which is fine. It gives more detail, but for teaching purposes and for my brain, I don't quite like it so well. And then I put another one in here. If you want to look at it side by side when you're studying, you have that option too. All right, but let's look at this. So this is just trying to show that when you have that phosphoglycolate, it's got to go through this big series of reactions. And there is some good news at the end, but let's work through this a little bit. So we have 3 different organelles that are involved. So that's pretty complicated. It starts off in the chloroplast, of course, but then certain compounds have to shift to the peroxisome and then to the mitochondria and then back to the peroxisome, then back to the chloroplast. So it's a lot of effort. And here, if you start in the top left in the chloroplast, you see RUBP and oxygen reacting. So you have to imagine Rubisco is right there. All right, so the active site, oxygen comes in, RUBP comes in, Rubisco reacts those two, and you get, if you follow this swooping line off to the right, you get one molecule PGA, which is good. And that can immediately feed into the C3 cycle. So I drew that little circle saying that PGA can go right into the C3 cycle. Great, because then sugars can get made from that. The other product is, if you go back over to the left, the other product is phosphoglycolate. All right. So now that's that funky 2-carbon compound. So that phosphoglycolate has to go through this whole process all the way through those different organelles. And actually, we're going to find out that it has to do this twice. You have to have 2 phosphoglycolate molecules coming through this process in order to get enough carbons to finally make yet one more PGA molecule. So let's look at this a little bit. Now, when you're studying this, the best way to study this is to know which organelles. I will ask you to know which organelles are involved and the order. You know, that first is chloroplast, then you go feed into the peroxisome, then feed into the mitochondria. And then I will be asking for what molecules are getting transferred from one organelle to the next. All right, so in this case, we see that phosphoglycolate reacts and you get glycolate. So I will ask you, what's leaving the chloroplast and getting into the next organelle? You would have to know glycolate is the one that's going into the peroxisome next. And then you see a series of reactions, but eventually that glycolate is converted to glycine. So that's what I want you to know. Glycine gets made in the peroxisome and gets shuttled over to the mitochondria. So glycine is only 2 carbons. So here in the mitochondria, they're showing this interesting little reaction. We're not going to get into it, but what they're showing here, okay, if you look over here, here's the one glycine coming in from the peroxisome. But look over here. There's another glycine on the far right of the mitochondria there. So this, the artist drew it. They're trying to say you have to have 2 glycines here in the mitochondria. So essentially you have to run this twice in order to get 2 glycines in the mitochondria, because then that will give you 4 carbons, which is necessary to then finish off this last half of the cycle. All right, so you have glycine getting made in the peroxisome. It gets transferred to the mitochondria, but you actually have to do that twice because you need 2 glycines. And then by the time it leaves the mitochondria, you have a serine molecule. All right. Now, let's see. Serine is 3 carbons. So we went from a 2-carbon glycine, a 2-carbon glycine, so that's 4 carbon, but we only see a 3-carbon leaving. So that means we're missing a carbon, and that's where the CO2 is coming in. So carbon is being given, we're losing a carbon molecule in the form of CO2 in the mitochondria. So this is where they say there's a loss of carbon. When they talk about the PCO cycle, they say you're losing carbon. That's what they mean. CO2 is being given off in the mitochondria, and it just is given off out of the plant. All right. And so they don't see that ever being used back in the chloroplast. So they say it's a loss of carbon. So that's one downside, potentially, when you think about it. That's not so good. All right, so CO2 is given off in the mitochondria. That's pretty important. I want you to know that as well. So by the time this glycine and this glycine go through this little reaction, one of the carbons gets lost in the form of CO2. You're left with a 3-carbon serine. Serine leaves the mitochondria, goes into the peroxisome, and then you get this little reaction pathway, and you end up with glycerate. Now we're getting close, because that should sound vaguely familiar. Now glycerate can move back into the chloroplast. And you see here the last step, pretty important here. You have ATP needed. You're expending energy to get glycerate to the form of phosphoglycerate, PGA. Which is good. I mean, now you got another molecule, PGA, that can feed right into the C3 cycle. That's good. But in this whole process, you still lost out a little bit because you lost the CO2 molecule. So you respired that off. And then you had to push extra energy into this whole process. You had to expend some ATP. But at least you did get some little bit back for it. You did get another PGA molecule that can then go into the C3 cycle. So this entire thing that you're looking at, this is the so-called PCO cycle that phosphoglycolate has to undergo in order to recover a little bit of good from it, in the sense that, yes, you do get a PGA molecule eventually after going through this whole thing essentially twice. You do get a PGA molecule, which can feed into the C3 cycle. Okay, and where is, you also see NADH getting used in part of the pathway. So that's another thing to consider, that you're losing, reducing molecules is needed to be used in part of it anyways. So, okay, let's move ahead a little bit and think about this. So what we just described is that essentially you have to run that cycle twice to get enough carbons to eventually make a PGA molecule. And so you'll get that glycine and that glycine forming in the mitochondria, and eventually that will turn into, you do get a PGA molecule, which is 3 carbons, but unfortunately you lose a carbon in the form of CO2. So if you do the math, if you have 4 carbons and 3 are used for something good and 1 is given off, they say 75% of the carbon is recovered in the form of PGA. That's good. That's a good thing. However, you do get a loss of carbon in the form of CO2 being given off or being respired. So that's why they say it's photorespiration. So last session, we said Rubisco really can't tell the difference between CO2 and O2. It pretty much doesn't care. It'll take either one into the active site. So whatever Rubisco is seeing more around itself, that's what's going to win and come into the active site. It's a competitive thing between CO2 and O2. And so, you know, if you take some plants in the lab and you start working this in the lab and you start playing around where you try to manipulate the situation, like, okay, I want to try to make the plant do the PCO cycle. Under what conditions will that start to really get cooking along? And so they've been doing a lot of research on this. And what they find is that if you have a solution, an aqueous solution in equilibrium with air at 25 degrees Celsius, if you look at the dissolved gas ratio in that solution, you have a CO2 to O2 gas ratio of about 3:1, which is good. That means you have more CO2 in just a bulk water. You'll have more CO2 and when it's 25 degrees Celsius, you know, all that. Okay, so that's good. However, you can experimentally manipulate that ratio and see what happens to the plant in terms of, will it do the Calvin cycle or is it going to do the PCO cycle? Well, if you create a situation where you manipulate it so that the oxygen levels decrease, then that's going to favor the C3 cycle. If you're knocking down oxygen levels, then Rubisco will carboxylate. CO2 wins the competition, CO2 comes into the active site, and you get the Calvin cycle or the C3 cycle. However, if you tip the balance and say, I want to manipulate the situation so that there's more oxygen or somehow drop the CO2 levels, then Rubisco will see more oxygen around it. Oxygen wins, comes into the active site, and you get the PCO cycle kicking in as a result of that. Well, when you're a plant living your life and you start to experience increased temperatures around you, hot, sunny day, that is actually, and they did this experimentally, of course, that is actually going to favor oxygenation. Why? Well, pretty much simply because as you heat a body of water that has dissolved gases in it, the solubility, SOL stands for solubility, the solubility of gases start to decrease as you increase the temperature of that water. They want to bubble out of solution, so to speak. But oxygen comes out slower than CO2. And that's just the chemistry and physics of it. So oxygen is less affected by higher temps. Oxygen will stay dissolved in the aqueous solution, which means it can stay around Rubisco longer under high temps. So as the plant body is getting heated up, of course, the liquid in the body of the plant is getting heated up. CO2 starts to come out of solution, which means that it can't be pushed into the Calvin cycle. Oxygen stays in solution longer, and so now Rubisco is seeing oxygen. So under natural conditions, if you put a plant under high heat conditions, this starts to happen in their body. And so the outcome is that Rubisco just starts to see more oxygen around it, and oxygen has the competitive advantage now and can come into the active site of Rubisco. So we'll talk a little bit more about that because that has a profound influence on where certain plants are native and adapted to, and why we don't see certain plants in certain geographical regions of the world over others. So we'll get into that. It's really quite amazing. So let's just finish this other thought though here with the PCO cycle. So of course, plant physiologists have wondered for decades, why is Rubisco so, we call it a sloppy enzyme. It's sloppy. It can do both oxygenation and carboxylation. That's pretty weird for an enzyme to be able to have that kind of flexibility in the fact that it's not so picky. It can take both of those molecules into its active site. Well, if you think about when Rubisco evolved, it evolved back when photosynthetic organisms evolved billions of years ago. Now we're talking not in the plant world. So bacteria were the first organisms to do photosynthesis. And there are some, bacteria started to form and evolve on the planet before oxygen was even in our atmosphere back then. All right, so they hypothesized that rubisco, is found in these microbes. And so rubisco evolved during a time when there wasn't any oxygen. That wasn't even a consideration to think about, so to speak. And so you can imagine why those enzymes, it didn't, if there's no oxygen around it, that didn't have to, try to tighten up, so to speak, its binding affinity, because oxygen just wasn't there. And so they point to that idea that oxygen or rubisco has been around, well before oxygen came into our atmosphere. And just to make a note, there are plenty of microbes out there that can do photosynthesis in the absence of oxygen. So they do it differently than what plants do. And they were here first, long before cyanobacteria that do it more like plants do. And so there's plenty of microbes that don't need oxygen, don't make oxygen when they do photosynthesis. All right. Well, then they've also looked at the pathway, the whole PCO cycle, and you look at the complexity of it, 3 different organelles and ATP's expended and CO2's given off. And it's like, wow, why doesn't rubisco tighten up over the years of evolution? And they said, well, maybe there's an advantage to doing the PCO cycle sometimes. Maybe it's a good thing. Otherwise, you think it would be phased out over the process of natural selection and adaptation. And so you look at all the intermediates in the PCO cycle, and they said maybe some of those intermediates are necessary at key points in the plant's life, and those get funneled into other reaction pathways that are important. And so they've looked at that, and they still are looking at all this. And then some scientists say maybe it acts as some kind of safety valve for dissipation of excess ATP, which this one I'm having a harder time believing. I'm not so sure about this one. And so, but they claim that there might be something to this and that it might, in kind of a convoluted way, protect the plant against photoinhibition. And I've read the paper on this and I'm still not convinced. I can't wrap my head around it quite to the point where I go, yeah, that's plausible. I mean, they need more data in my opinion, but anyways. So this area of research is still really active because they're trying to genetically engineer, so to speak, rubisco and tweak it to try to tighten it up in terms of its binding affinity. Because experimentally, if you take plants and run experiments, well-designed, controlled experiments, if you force a plant to do the PCO cycle a lot, you can see that the yield of whatever, the growth, essentially, the plant growth starts to tank and it gets stressed easier. And so a lot of people that are trying to grow crops to feed billions of people, they're like, well, one way we could, grow higher yields is if we could reduce how much these crops are doing the PCO cycle. So that's kind of their goal, is if we can just produce more crops, one way we could do that is make rubisco more efficient. And then we could get higher yields, better crop production. And so they're definitely looking at this. This is a really active area of research, but and there's a couple of groups that are making some strides. Actually, I'll post the paper, not for mandatory reading, but if you're curious, there's some interesting research going on with this. Okay, so there's kind of a mixture depending on who you talk to. Some scientists say we need to get rid of this sloppiness of rubisco, and then other scientists are like, well, let's take a breath here. Maybe that's not the best idea. And so it's, like I said, there's a lot of dynamic interactions amongst the scientists on this topic right now. But there are several groups that are getting into, let's try to make rubisco more efficient or somehow work it so that the Calvin cycle is definitely being favored. Okay, so lots of stuff to see about that over the next few years. But some plants have come up with their own solution. And this is also a really active area of research because these plant physiologists are studying this, saying, if we can study these plants who did it all on their own and figured out a way to kind of outwit rubisco, maybe we could take what they're doing and put it into plants that aren't doing it yet. And so a lot of, again, tweaking of pathways and genetic modification that they're talking about. So it's a real interesting field of research. So here's a group of plants that we'll talk about first, the C4 plants. And the cycle, it's called the C4 cycle. C4 is just referring to the fact that there's 4 carbons in the first stable molecule in this cycle. And when you look at where these plants are native or where they originally adapted to live, it's tropical areas or subtropical areas, not around here. We don't have C4 that are native to this area. They can be grown here, but they're not native to here. When you look at them under the microscope, one thing that they noticed right away with the limited technology they had decades ago is that the leaf anatomy is physically different compared to plants that we call the C3 plants, like a typical plant around here is a C3 plant. How is it different? Well, all plants have a bundle sheath around. So a bundle sheath is just a ring of cells around the vascular tissue in the veins. And I'll show you a picture. But in the C4 plants, the bundle sheath, those cells are really plumped up. They're really big, really plump, and lots of chloroplasts. So we'll look at those. And then their mesophyll design, the way they arrange their mesophyll cells is also different compared to just a straight ahead C3 plant. So we're also going to see that when they run their Calvin cycle and the C4 cycle, it's going to be a spatially different area in the leaf tissue that we see these things happening. And I'll explain more. But don't forget, C4 plants definitely have a C3 cycle. I like to think of it as all plants have a C3 cycle. That evolves first. That's the Calvin cycle. All plants have that. It's just some plants have adapted like extra cycles to tack on to this. All right. And so the C4 plants, I like to think of it as the C4 cycle is kind of tacked on to the front end of the Calvin cycle to modify it so that it's really favoring the Calvin cycle to keep going and to minimize how much the PCO cycle runs. So when you look at the tropical and subtropical conditions that these plants are exposed to on a daily basis, they often have hot sunny days and periods of drought. And so these plants, when you experimentally put them under these conditions and compare them to just straight ahead C3 plants, the C4 plants win when you put them under consistently hot, sunny conditions, and you stress them a little bit with drought. C3 plants tank, C4 plants are like, yeah, I'm fine. All right. And so they have adapted to these types of conditions, and that C4 cycle is the reason why they can do it so much better. And so experimentally, they can say, 2 to 3 times the photosynthetic rate these C4 plants can do over the C3 when they put them head to head in carefully controlled experiments. So when you look at it and you examine the C4 cycle and you look at it in totality and you say, what's going on here? Why did this evolve? And if you really look at it, you realize that it's a way for them to concentrate the CO2 right around rubisco. And I like to think of it like rubisco's taking a bubble bath, in a big clawfoot bathtub. And all those bubbles are the CO2 that the C4 plants are just pushing at rubisco. And it's just submerged in all these lovely CO2 bubbles, so to speak. So whatever you have to do to help you think about what's going on with this, imagery is a good thing. So it's concentrating the CO2 around rubisco when you look at the C4 cycle. So let's work through this a little bit so I can convince you of that. First, let's look a little bit at the anatomy. So here is a C4 leaf under the microscope, and I think this is a corn leaf. So you're looking at the leaf in cross section, and so these areas here where you see the pink and then the kind of blue-green cells, here is a really big one, the pink and the blue-green, that's the xylem and the phloem. All right, the xylem are the red, phloem are the blue-green. The bundle sheath cells are these big plump cells that surround. So the bundle sheath is a ring of cells that surround the veins. Okay, so this is a major vein. You can see big, plump bundle sheath cells. Here's another smaller vein, bundle sheath cells, bundle sheath cells. Okay, so nice and plump. Let's compare that to a C3 plant. Here you can see they have bundle sheath cells too, but they're not nearly as well developed in terms of their physical size, and you don't really see a bunch of chloroplasts in there. Here's another image of a C4, and this one's nice because they've stained the chloroplasts quite nicely. So you can see all the kind of bluish dots, those are chloroplasts in the bundle sheath cells. So you have lots and lots of chloroplasts. The bundle sheath cells are nice and plump. So that's one big difference when you look at a C4 and a C3 plant. Then the other thing too, in this drawing, maybe we'll show it a little easier. Up top, you see a C4 plant. And the artist is trying to show how the spongy mesophyll cells, look how they hug around where you have the veins. All right, so they're a little bit more closely associated with the bundle sheath cells. I guess that would be the best way to show it. So here the green or bluish green, I guess it's blue, is the bundle sheath, the way the artist drew it. And then you see these mesophyll cells that are hugging up to the bundle sheath. And so there's a little bit more just physical intimacy and connection between the bundle sheath and the mesophyll. Whereas if you compare down here in a C3 leaf, you can see the spongy mesophyll are just kind of scattered, loosely associated with the bundle sheath. So that's another physical difference. And that becomes important because we're going to see how certain parts of the C4 cycle happen in the mesophyll, certain parts happen in the bundle sheath, and it makes sense to have them hugging up next to each other so you can get transfer of these molecules back and forth as the cycle cooks along. So when they look at these mesophyll cells and the bundle sheath cells, lots of plasmodesmata connections between these cells so that you can get nice transfer. So as the C4 cycle is cooking along, you can get the transfer through the plasmodesmata, and it's very efficient that way. It's just an open channel going back and forth between the two types of cells. So here's the drawing of that idea. So the top cell is the mesophyll cell, and then the bottom cell here is the bundle sheath. All right. So this is, again, kind of the overview of the C4 cycle. And you can see they broke it down in different parts. So the first part is carboxylation. And that's also what happens in the C3 cycle. So here you're seeing the CO2 coming in to the leaf, getting to the mesophyll cell, and you see instead of it reacting with RUBP, you don't see that. Instead, it's reacting with phosphoenol pyruvate, which you can abbreviate down to PEP. So RUBP is not even there. It's not there. All right. So when CO2 comes into a C4 plant, it's not going to react with RUBP right away. Instead, it'll react with phosphoenol pyruvate, and it forms a C4 acid, which is oxaloacetate, and I'll show that on the next slide. And then you can see that, well, and then it converts to malate. I'll walk you through all this. And then malate will come into the bundle sheath through the plasmodesmata. It's 4 carbons. And then you see a decarboxylation step. And in the bundle sheath, that's where you see the Calvin cycle. All right, so the artist put the Calvin cycle in the bundle sheath. It did not, whoever drew this knows it does not belong in the mesophyll. All right, so the Calvin cycle is happening in the bundle sheath cells in C4 plants. So you have this decarboxylation step where that C4 acid, which typically it's malate, will get a CO2 cleaved off of it. And then they're trying to show that that CO2 can feed right into the Calvin cycle. So this is where Rubisco is. Rubisco is in the bundle sheath cell. It's not in the mesophyll cells. It's in the bundle sheath cells. And if you can decarboxylate in the bundle sheath and push all that CO2 around Rubisco, well, then it's going to see a lot of the CO2. That's what it's going to have high amounts around it, and it will win the competition to come into the active site. So that's great. Well, when you take a CO2 off of malate, you're left with 3 carbons. So that 3 carbon acid will have to move back into the mesophyll cell to regenerate. And it's trying to regenerate the acceptor molecule, which is PEP in this case. So this is the big picture idea of the process. So with C4 plants, carboxylation is the first step, but it's not with RUBP. That's not what's getting carboxylated. It's PEP instead, PEP. Then you have to shift over physically to a different site in the bundle sheath and decarboxylate. So really, I like to think of the C4 acid, which is typically malate or malic acid. I tend to think of malate like a vector. You know, when you talk about vectors that will transmit, like the mosquito is a vector that transmits malaria to people, right? And so we say that the mosquito is a vector of the plant or the human pathogen. So I like to think of malate as a vector for CO2. It's moving CO2 in the form of malate from one physical location to another. And then it serves its purpose. It's like, okay, I've brought CO2 as part of my malate molecule, and now I can decarboxylate and push that CO2 into the Calvin cycle, right where Rubisco is. And so it's just a very efficient way to get CO2 to get physically from point A to point B. If you put it part of a molecule, that molecule is dissolved and can get easily transferred. So it's a nice little strategy. Okay, so in words, so if you're more of a read-write person, in words, what we're talking about from that previous slide is here. So when you run the the C4 cycle, the first stable carbon compound is oxaloacetate, OAA. You can abbreviate that down to OAA. So that's when CO2 comes in, and when CO2 dissolves in water, then it becomes a bicarbonate. So HCO3 minus is often how they will show the CO2. Reacts with PEP, and you get the oxaloacetate. What enzyme? It's not Rubisco enzyme. Instead, it's a totally different enzyme that we call pepcase or PEP carboxylase. So carboxylase, it's still working with the CO2. It's grabbing CO2 and bringing it into its active site, but it will react it with PEP. Pepcase is a wonderful enzyme in the sense that it has a really high binding affinity for CO2. It will not take oxygen into its active site. So there's no competition. This enzyme, it will take CO2 and react it with PEP, and that's it. And so that's good. You don't have this wishy-washy, sloppy enzyme creating issues. And when they do the tests in the lab, the biochemists will say, yep, this enzyme's really efficient. And another way to say that is that it's saturated, its active site is saturated nearly all the time. And you can run the kinetics of it, really great kinetics in this reaction. So that's good. And oxygen is not a competitor. It only takes in CO2 into its active site, and then we'll take, and react it with PEP. So that's great. When you have that carboxylation step with C4 plants, it's very efficient. And you end up with oxaloacetate, which then gets converted, rearranged to malate. It doesn't lose a carbon or anything. It just gets rearranged into malate. And then it's malate that I think of as the vector, because it's going to physically move now and shift over to the bundle sheath cell, BSC, bundle sheath cell. So now let's look at the C4 cycle in a little bit more detail compared to that previous image, and we'll work through this a little bit. All right, so here we are with the top cell again being a mesophyll cell and the bottom cell being the bundle sheath. So let's look for where CO2 is coming in. Here it is up top. You have the CO2 coming in and reacting with phosphoenolpyruvate. So you can see that's a 3-carbon compound, PEP, reacting with CO2. So then you get a 4-carbon oxaloacetate. So that's why they call it the C4 cycle. And then you can see the formation of malate. right after that. So you do see NADPH is necessary. So you do have to use a reducing molecule, which is kind of precious. So there's that issue. And then we'll talk about ATP being used as well. So here's malate, 4 carbons still, all right? And you see malate acting as a vector, so to speak, shifting from the mesophyll into the bundle sheath. Then you got to get that CO2 that's part of the malate molecule off of malate. So you have to decarboxylate next in order to cleave off that CO2 and feed it right into the Calvin cycle. So here in the bundle sheath, that's where we see the Calvin cycle. That's where Rubisco is. Now CO2 is getting fed to Rubisco, and the Calvin cycle can cook right along and make all that lovely sucrose eventually. If you take CO2 off of malate, you're left with pyruvate, a 3-carbon compound. So pyruvate will shift back into the mesophyll cell, and then here you finally see, okay, now we have to expend some energy to do this last step of taking pyruvate and forming phosphoenolpyruvate off of that. So there's a cost to this C4 cycle. There's a definite cost. However, the benefits must outweigh the costs because there are many C4 plants when you look at plants that are native to tropical and subtropical areas. So it must be worth it. Okay, so let's, in words, so the carboxylation, we talked about that in the previous slide in words. Now let's do the decarboxylation in words. So that's happening in the bundle sheath. And that's where Rubisco is. So once malate comes into the bundle sheath and delivers the CO2, essentially, the decarboxylation is cleave the CO2 off, get it into the Calvin cycle or the C3 cycle, but then you got to shift the pyruvate back to the mesophyll cell to finish the C4 cycle. And once you get the pyruvate over back into the mesophyll cell, then you do the third step, which is regeneration. And that's where you take pyruvate and you finally get your PEP acceptor molecule back. It's a funky enzyme. You don't need to know the name of the enzyme, but if you're curious, there it is. It's a funky enzyme that actually takes ATP and has to cleave 2 phosphates off of it, in order to push this reaction. And that's why you see the adenosine monophosphate. Usually you only see 1 phosphate coming off and you get ADP, but this time it's AMP. So it's kind of a funky little thing here. So overall, what do we see? If you compare the C4 cycle to the C3, it's 2 extra ATP. And so in total, if you have 1 CO2 coming in, you'll have to expend in total to do the C4 and C3 cycle, the 5 ATP and 2 NADPH. JZ, did you have a question? I thought I saw your name.
Yeah, I was going to ask, why doesn't it just use 2 ATP molecules instead of you cleaving 2 phosphates off of 1?
Good question. And I do not know the answer to that. I've wondered that.
Then I was just going to say it's probably just because evolutionary selective pressure. So.
There's some kind of selection pressure that favored this over what you described, and I don't know why. It'd be fun to take a time capsule back and see how this all happened, but unfortunately we can't do that. So, okay. So we've looked at it in visual form, wrote it all down here. There's a little bit, some detail I want to impress on you though. There's a little quirk here that we need to think about. All right, so I created this table to try to parse out where everything is happening. So the fixation or the carboxylation, I probably should rename that carboxylation instead of fixation. It means the same thing, but your figures are showing it as carboxylation. So the carboxylation or fixation is the CO2 coming into the mesophyll, reacting with PEP with the peptase enzyme, forming OAA, which then forms malate. That's all happening in the mesophyll. Then the malate shifts to the bundle sheath cell, and you get the decarboxylation event where CO2 is cleaved off of malate, and that can go right in. So the CO2 goes right into the C3 cycle. So the C3 cycle is happening in the bundle sheath cells. Pyruvate will shift back to the mesophyll cell to get your acceptor molecule, PEP, up and ready to go to take in, to react with more CO2. Okay, and so I'm delineated out where this is all happening. Okay, so now let's think about it for a second. In the C3 cycle, when we think back to what we were talking about with the C3 cycle, and we look at it. So in the C3, you have CO2 and RUBP reacting, and you get your 2 molecules of PGA. And that all just happens spontaneously. Then the next step of the C3 cycle is PGA gets converted to G3P. Now that part of the C3 cycle, you do have to expend ATP and NADPH. So C4 plants have tweaked the C4 cycle where, actually they've tweaked the C3 cycle, I should say, because what they have done to further protect Rubisco against seeing too much oxygen is they shift the PGA will get exported instead of the C3 cycle all happening in the bundle sheath cell. What they do is once PGA is made in the bundle sheath cell, they'll physically push PGA, so to speak, over to a mesophyll cell so that it's over in the mesophyll cell that we can see PGA getting reduced to G3P. Now, that sounds like an extra, like, picky little detail. Like, why would the plant do that? That just seems weird. But when you look at the Calvin cycle and you look at the formation of G3P, so you go from PGA to G3P, you look at it and you go, oh, well, you need ATP to do that. You need NADPH to do that. Where's the plant getting all this lovely ATP and NADPH? What process?
The light reactions?
Yes, thank you. The light reactions. All right, so the plant that's where it's getting the lion's share of ATP and NADPH. It's cranking out the light reactions. Well, now we finally have to think about oxygen. When we first talked about the light reactions, they said, yeah, ignore the oxygen that gets made. Well, now we do have to think about it. So as the light reactions are cooking along, one of the other products is oxygen. And we know Rubisco can't tell the difference between oxygen and CO2. So this little tweak that the plants do to kind of split where the C3 cycle is happening is, again, it's done to make sure that Rubisco really isn't seeing much oxygen. If you shift and do the reduction phase of the C3 cycle away from Rubisco, shift it over to the mesophyll cells. Let the mesophyll cells take care of that part, and that's fine. And then once that part is done, you can bring back the G3P back into the bundle sheath cell and finish the Calvin cycle. So C4 plants not only have this amazing C4 cycle that they've tacked on to the C3 cycle, they've also split where the C3 cycle happens to further reduce the option of Rubisco seeing too much oxygen. And so that means the PCO cycle will be suppressed. And the C4 plants are amazing at suppressing the PCO cycle, making sure that Rubisco is seeing way more CO2 than oxygen. Okay, so when you think about that, going back to PGA, when they shift the PGA over into the bundle sheath, this is what's happening. Okay, this is the reduction phase of the C3 cycle. So C4 plants say, okay, I'm going to push PGA out to the mesophyll cell. Let this happen in the mesophyll cell because it's during that stage that you have oxygen being produced to get ATP. You have oxygen being produced to get NADPH because it's all happening during the light reactions. And part of the light reaction product is oxygen. And so stick that out into the mesophyll cell. And then once you get G3P made, that can come back in and be used to finish off the C3 cycle in the bundle sheath. So it's a little gnarly, a little complicated, so please spend a little bit of time thinking that over. Your book does discuss it too, as well. But we have to stop there. So we're gonna talk, just wrap up C4 plants next time. And then we'll also get into another adaptive strategy to suppress the PCO cycle with the CAN plants. We'll do that next. Yes,
Is our exam still on schedule, as scheduled, or is it already behind?
for the next exam. Oh. So the next exam, exam 3 is on the syllabus for week 11, which is 2 weeks from now, correct? I think. So probably will be on schedule. And it's going to be heavily focused on the light reactions for sure. So that will be the super main topic of exam 3.
I also had a question about exam 3.
Yeah.
I was just wondering, sorry, can you hear me?
Yes, I can.
Okay, I was just wondering, if like for the written questions, can you put like things where we can attach pictures? Because this is like something similar we were learning in medicinal plant chemistry, and he let us like write down this pathways before we like wrote the paragraph about it. Is there a chance you could do that? Because I just think it's easier to write it out before I can like actually put it into words, you know?
So you'd like to be able to like take pictures? Is that what you're saying?
Yeah, so I could
Sketch
like
it out
write
on a
out
piece
the
of paper.
yeah, write it out on a piece of paper and then also explain it in words.
Okay, let me think that over and we'll see what we can do to maximize success, okay?
Okay, thanks.
Any other questions?
I had something about my research project.